Okay, good afternoon, everyone.
Good afternoon, ma'am.
Good afternoon. So, in the last class, we had discussed about proteins, right? Proteins. Some common ideas about proteins, then the classification of proteins. Then, now we are going to learn about the structure of protein. Okay, structure of protein. Structure of proteins. This we learned that this alpha amino acid, they are considered as the monomer of this protein, right? So, proteins are the polymers of alpha amino acids and they are connected to each other by peptide bond or peptide linkage. I told you amino acid units, they are joined together to form a large single unit that is called protein, right? So, here these alpha amino acids, they are joined together by peptide bond or peptide linkage. The bond between two amino acids, it is known as peptide bond or peptide linkage. This peptide linkage is an amide formed between COOH group and NH2 group. The elimination of a water molecule and formation of a peptide bond, that is CONH. So, this peptide linkage is a amide formed between COOH group and NH2 group. You know that there are two groups there, right? NH2, that is amino group, then COOH, right? And there is one water molecule will be removed from this, and here it's formed CONH. Okay, CONH. From here this OH and here one H is eliminated, then CONH. This one is known as amide. So, this bond is known as peptide bond or peptide linkage. Clear? Proteins are the polymers of alpha amino acids. That means alpha amino acids, they are joined together to form a large polymer. It is known as protein. And these small amino acids, they are joined together by peptide bond or peptide linkage. This peptide linkage is the formation of an amide between carboxylic acid group and amino group. From this carboxylic acid and amine, one H2O molecule is eliminated. The remaining portion is known as amide, CONH. If you write it like this, it will be more clear. COOH, then NH2. Okay. Then these molecules are removed, then you can join CONH. This one is known as amide. Okay, this one is known as amide. This is clear? Yes. Then the product of the reaction is called dipeptide, because two amino acids, they are joined together. It is known as dipeptide because it is made up of two amino acids. You can look at the example here. These two are the examples of two amino acids, and from these two amino acids, H2O molecule, it is removed. Okay. Yeah.
Can you draw on the diagram? Like we can't understand which one you're explaining.
Okay. Just wait. NH2, then CHR, COOH. This is one amino acid, right? This is one amino acid. Then it is joined together with another one. Here also one COH group, then HR. Here one NH2. Okay, these are two amino acids. So, one H2O molecule is removed from this. From the first one OH and the next one hydrogen. So, H2O molecule is removed from these two amino acids and the formation of CONH take place. Okay, formation of CONH take place. This is an example for a dipeptide. That means two amino acids, they are joined together by peptide bond or peptide linkage. It is known as dipeptide. So water molecule will be removed from these two amino acids, then amide is formed. Okay, then you can join these two, CO, then CN. Here you can see the bond is formed between these two amino acids. Is this clear now? Yes. So proteins, they are the polymers of alpha amino acids and they are connected to each other by peptide bond or peptide linkage. This peptide linkage is an amide formed between COOH group and NH2 group. The elimination of a water molecule and formation of a peptide bond, that is CONH. CONH. The product of the reaction is called a dipeptide because it is made up of two amino acids. Have you completed this slide, children?
No, ma'am.
Yeah, then you can complete it. Okay, finish, right? Okay, then if third amino acid combines to a dipeptide, dipeptide means it's the combination of two amino acids. If one more amino acid is linked to this dipeptide, it is called tripeptide. Okay, it is called tripeptide. So here three glycine amino acids, they are joined together to form a tripeptide. This is the structure of glycine. Okay, NH2, CH2, COOH. So from here one OH, then from here one hydrogen. It's removed. then it is linked to one more amino acid. Only water molecules are eliminated from this reaction, and here 3 amino acids are joined together, so it is called a tripeptide. Here it is number of, okay. When the number of amino acids is more than 10, then the products are called polypeptides. The number of amino acids joined together to form a protein that is more than 10, it is called polypeptide. A polypeptide with more than 100 amino acid residues was having molecular mass, molecular mass higher than 10,000 U, it is called a protein. A polypeptide with more than 100 amino acid residues. That means more than 100 amino acids, they are joined together by this peptide bond with an elimination of water, and their molecular mass, it is higher than 10,000 U. U stands for atomic mass unit or unified mass. It's the unit of atomic mass. So 10,000 U, it is called a protein. So more than 100 amino acids joined together with a molecular mass of 10,000 U, it is called a protein. This is clear?
Ma'am, can you say it once more?
Sure. A polypeptide means more amino acids, that means more than 10 amino acids, they are joined together. So here in the case of proteins, more than 100 amino acids, they are joined together with a molecular mass higher than 10,000 U, it is called a protein. Otherwise it's simply dipeptide, tripeptide, tetrapeptide, like that. Okay, if there are more than 100 amino acids and their atomic mass is more than 10,000 U, it is called protein. Not 1000, 10,000. It is called a protein, that's it. Clear, Vidu?
Yes, ma'am.
Okay. And this distinction between polypeptide and protein, it is not very sharp, because in certain cases with 51 amino acids, okay, here in its definition, a polypeptide with more than 100 amino acid residue, it is called a protein, right? But sometimes with at least 51 amino acids, they are joined together to form a polypeptide, it will also considered as a protein. Insulin is such a protein with 51 amino acids. So the distinction between this polypeptide and protein, it's not very sharp. Clear? So you can write one exception here, 51 amino acids joined together, it is known as insulin. It's an example for one protein. Have we completed?
Yes, ma'am.
All of you? Okay, then these proteins can be classified into two types based on their molecular shape. Okay, proteins can be classified into two types on the basis of their molecular shape. First one is fibrous protein. Fibrous protein. When the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then the fiber-like structure is formed. When the polypeptide chains, you know that this more than 100 amino acids, they are joined together with peptide bonds to form this protein, right? So this polypeptide chains, that means more amino acids there. These polypeptide chains run parallel and are held together by hydrogen and disulfide bonds. So this is the linkage between these chains, hydrogen and disulfide bonds. Then a fiber-like structure is formed. These fibrous proteins, they are insoluble in water. Insoluble in water. Then the examples for such fibrous proteins, keratin. It's present in hair, wool, silk, etc. Next one is myosin. It's present in muscles. These two are examples of fibrous proteins. So they are parallel structure and held together by hydrogen bonds and disulfide bonds. So here a fiber-like structure is formed and it is insoluble in water. Have you completed?
Okay.
Okay. Finished, right? Yes. All of you, Vidur?
Yes, ma'am.
Okay. The next type is globular proteins. Second one is known as globular protein. This structure results when the chains of polypeptides coiled around to give a spherical shape. So in the case of fibrous protein, they are parallel in shape. Then they are similar to fibers, right? Here the structure result when the chains of polypeptides, they coiled around and got a spherical shape. And this one is soluble in water. Okay, this one is soluble in water. And the examples are insulin, albumin, etc. These are the examples for globular proteins. Insulin, then albumin, etc. Adul, which are the two types of proteins depending upon their structure?
Fibrous and globular.
Yeah, fibrous and globular proteins. Gayatri, can you tell me examples for fibrous proteins?
Ma'am, I couldn't write that actually.
Okay, okay. Aditya, can you tell me? Fibrous.
Ma'am, keratin.
Yeah. Gayatri, you can write this. It's keratin present in hair, wool, silk, and myosin. It is present in muscles. These are the examples of fibrous proteins. Vidur, what about globular proteins examples?
Insulin and albumin.
Yeah, insulin and albumin. Completed?
Yes, ma'am. Thank you.
Yes. Then what about this slide? Finish, right? Then we are moving to the structure of these proteins. Okay? There are 4 different levels. There are 4 different levels for the structure of protein. First one is primary, then secondary, tertiary, quaternary. Primary structure is there, then secondary structure is there, tertiary and quaternary. Then we can discuss each one in detail. Structure and shape of proteins, it can be studied at 4 different levels. First one is primary, next one is secondary, then tertiary, and the fourth one is quaternary. Children, switch on your videos. Then we can discuss each one in detail. First one, primary structure of proteins. Okay, primary structure of proteins. Proteins may have one or more polypeptide chains. Polypeptide chains means the amino acids more than 10 they are joined together to form polypeptide chains. So in the case of protein, they may have one or more polypeptide chains. Each polypeptide in protein has amino acids linked with each other in a specific sequence, and it is this sequence of amino acids are said to be the primary structure of protein. That means polypeptide chains, they are joined together to form a particular structure in a straight way. That is known as primary structure of protein. Clear? Proteins may have one or more polypeptide chains, and each polypeptide in protein has amino acids. You know that monomer of this protein, it is considered as amino acid. So each amino acids, they are joined together to form this polypeptide, and they follows a specific sequence of amino acids, and it is said to be the primary structure of protein. Primary structure of protein. At the one end there will be one amino group, and the other end there will be one carboxylic acid group. The reaction is same, peptide bond is formed, that means elimination of water take place. Okay, so this amino acids, they are joined together to form this polypeptide. So one or more polypeptide chains present in proteins, they follow a particular sequence with the elimination of water with peptide bond or peptide linkage. This is the primary structure of protein. Michael, Meenakshi, Muhammad Adim, Srihari, Naurin, Rijaz, Evelyn. All of you switch on your videos. Steffi, can you please read the points about primary structure of protein?
Yes, ma'am.
Yeah.
Proteins may have one or more polypeptide chains. Each polypeptide in protein has amino acids linked with each other in a specific sequence, and it is this sequence of amino acids that is said to be the primary structure.
Yeah, primary structure of protein. Okay, then we are moving to the, have you completed? Yes, right? All of you? Yes, then we are moving to the secondary structure of proteins. Okay, secondary structure of proteins. The secondary structure of protein, it refers to the shape in which a long polypeptide chain exists. So the secondary structure, it will provide the shape of this polypeptide chain. It found to exist in two different types of structures. First one is alpha helix. Okay, there are two possibilities here, alpha helix and beta pleated sheet structure. Beta pleated sheet structure. First one is alpha helix and the second one is beta pleated sheet structure. Okay, the secondary structure of protein refers to the shape in which a long polypeptide chain exists. It found to exist in two different types of structures, alpha helix and beta pleated sheet structure. It arises due to regular folding of the backbone of polypeptide chain due to hydrogen bonding between carbonyl group. Okay, in the primary structure I told you it's a straight chain with proper sequence, right? In the secondary structure of proteins, there will be one folding. Okay, this folding related to the bond between the hydrogen bond between carbonyl groups. Due to the bonding between the carbonyl group, there will be one folding in that straight chain. That is known as secondary structure, and it will be exist in two forms, either alpha helix structure or the second one is beta pleated sheet structure. CO and NH bonds. Okay, hydrogen bond between CO and NH bonds, not bonds, NH groups. That means amino and carboxylic acid groups. Then these two are the images of alpha helix structure and pleated sheet structure. Alpha helix is in a zigzag motion, then beta pleated, it is just parallel with folding.
Ma'am,
Yeah.
is it polypeptide chain due to hydrogen bonding between
CO and NH groups. Mehak, can you please read the points about secondary structure of proteins?
The secondary structure of proteins refer to the shape in which the long polypeptide chain exists. Found to exist in two different types of structures, alpha helix and beta plated sheet structure.
Yeah.
That is due to regular folding of the backbone of the polypeptide chain due to hydrogen bonding between CO and NH groups.
Yes. Carbonyl groups and amino groups. Finished? Then we are moving to tertiary structure. Okay. Tertiary structure of proteins. Overall folding of the polypeptide chains, that is further folding of secondary structure. I told you in the primary structure it's a straight chain. In secondary structure the folding of that polypeptide chain. Then here in the tertiary structure one more folding of secondary structure. That gives tertiary structure of proteins. Clear? Overall folding of the polypeptide chains, that is further folding of secondary structure. It will give tertiary structure of proteins. It gives rise to two major molecular shapes, fibrous and globular. We learned about two types, right? So from the tertiary structure we can able to distinguish fibrous protein and globular protein. Okay, give rise to two major molecular shapes, fibrous and globular. Then the forces which stabilize the secondary and tertiary structures of proteins are hydrogen bonds, disulfide linkages, van der Waals and electrostatic forces of attraction. So these are the different forces that exist between the molecules of amino acids or polypeptides. Which are they? Hydrogen bonds, disulfide linkages, van der Waals and electrostatic forces of attraction. That will stabilize the secondary and tertiary structure. It's a folded form, right? So in order to maintain its folding, these type of forces, they help us. Hydrogen bonds, disulfide linkages, van der Waals and electrostatic forces of attraction. Primary structure it's already stable because straight chain, right? In the case of secondary it's a folding of straight chain. Then in tertiary one more folding there. So in order to make it stabilized, certain forces are there. Hydrogen bond, disulfide linkage, van der Waals and electrostatic forces of attraction. Switch on your videos, children. Anna, can you please read the points about tertiary structure?
Tertiary structure of protein, overall folding of the polypeptide chain, that is further folding of secondary structure,
Okay.
gives rise to two major molecular shapes, fibrous and globular.
Yes.
Forces which stabilize the 2' and 3' structures of proteins are hydrogen bonds,
Secondary and primary structures.
secondary and primary structures of proteins are hydrogen bonds, disulfide linkages, van der Waals and electrostatic forces of attraction.
Okay. Finished? Then the last one, that is quaternary structure of protein. Quaternary structure of proteins. Some of the proteins are composed of 2 or more polypeptide chains referred to as subunits. So here in the quaternary structure, it's the spatial arrangement of these subunits with respect to each other. It is known as quaternary structure. It is more complicated compared to the primary, right? Here the spatial arrangement of this folded structure of polypeptide chain, that is known as the quaternary structure of protein. Okay. You know that polypeptide chains, they are joined together to form this proteins, right? Polypeptide chain means more than 10 amino acids. They are joined together with peptide bond. So here the spatial arrangement of the subunits with respect to each other, it is known as quaternary structure. Okay. Quaternary structure of protein. In this figure, each colored polypeptide chain, that means there are 4 polypeptide chains here in this protein. Okay, that's why they are shown in different color. That's the quaternary structure of protein. So these 4 are the structures, 4 levels of structure of protein. Then we can also need to discuss the alpha helix and beta plated sheet structure from the secondary structure that we can discuss in the next class. Then one more topic in this protein, that is the denaturation of protein. Okay, that also we can discuss in the next class. Okay, so is this clear, children? We just started from structure of proteins. So you have to remember peptide bond or peptide linkage is here. Alpha amino acids, they are joined together to form this proteins. And in this peptide linkage, water molecule is eliminated from this amino acids. Then dipeptide, here 2 amino acids are joined. Then tripeptide, 3 amino acids are joined. In each joining, that means elimination of water molecule and peptide bond is formed. Amide is formed between these two. Okay, with the elimination of water, amide is formed. Then if there is more than 10 amino acids, we called it as polypeptides. Then polypeptides with more than 100 amino acids and their molecular mass is greater than 10,000 atomic mass unit, it is called a protein. But it's not a sharp distinction between these two. The 51 amino acids consist of insulin. It's also an example for protein. Okay. So there are certain exceptions there. Then there are 2 types of proteins depending upon their shape, fibrous protein and globular protein. This keratin and myosin is an example for fibrous protein. Then for globular protein, insulin and albumin. Then we learned the 4 levels of structures of protein: primary, secondary, tertiary, quaternary. The primary structure, it's just a straight chain with peptide bond linkages, and in the secondary structure it's a folding of primary structure, and it is in 2 forms: alpha helix and beta pleated sheet structure. Then the tertiary structure, it is overall folding of this polypeptide chains, and it gives rise to 2 types of proteins according to their shape, fibrous and globular. Then there are certain forces that stabilize secondary and primary structures. They are hydrogen bonds, disulfide linkages, van der Waals, and electrostatic forces of attraction. Then quaternary structure of proteins. That is a spatial arrangement of these subunits, polypeptide chains. That is quaternary structure. The next class we can discuss about alpha helix structure and beta pleated sheet structure along with denaturation of proteins. Okay, this is clear? Yes or no?
Yes, ma'am.
Yes, okay. Then, okay, now it's time to wind up. So thank you all. See you in the next class. Bye.
Thank you.
Thank you.